Okay. Well, thank you everyone for joining today for the institutional seminars of the League. So today I have the pleasure of hosting and introducing Dr. Joris Veltman. Joris is a professor at the University of Newcastle in the United Kingdom and Dean of the Biosciences Institute of this university. He received his doctorate degree from the University of Maastricht in the Netherlands. And after a postdoc at USCF, returned to the Netherlands as a professor at Radboud University Medical Centre in Nijmegen, where he performed and directed studies focused on the understanding of the contribution of de novo genomic variation to human disease. He has received multiple awards and recognitions for his contributions to human genetics, including the Radboud Science Award, the King Faisal International Prize for Medicine, and the Royal Society Wolfson Research Merit Award. So thank you very much, Joris, for joining us today. And Joris is going to talk about de novo mutation in disease.
Thank you very much for the invitation, Claudia. Is my screen up okay like this?
Yes, we can see it.
Perfect. Well, thanks very much. It's a great pleasure to present to you all, to the Institute. Of course, it would always be much nicer to visit in person, but that's just very challenging, and online presentations are just very easy to organize. So it's really great to do these things, and I love to do that. So great to be now virtually in Mexico for an hour. So as Claudia said, I've worked for a long time at Radboud University in Nijmegen, and I've moved 5 years ago to Newcastle University. And my presentation basically will talk about work that I did during that period. So both universities I have to acknowledge here. And I'm mostly going to talk about sporadic genetic disease. So I hope that will be interesting. I'll give you a bit of an overview of what we've done and ask some questions and try to answer them for you. So hopefully that'll be entertaining. So when we classically think about genetic studies, we used to always think about these kind of large families where a disease is segregating through a family, and you would basically try to find rare inherited variants that would associate with disease. On the other hand, when you have, so this is for rare disease, but also for common disease, we would actually look at common variants that are associated with a disease in genome-wide association studies in a similar way. And from this, a lot of, you could imagine that if you think about genetics, you think, hey, actually, this is very much associated with variants that associate spread through the population and spread through families. However, the thing with genetic diseases is, especially with monogenic diseases, is that they often occur sporadic. And especially if these diseases occur early in life, and especially if they result in severe disease. So if you think about, for example, severe forms of developmental delay or intellectual disability, it's what I've studied most of my career. These diseases, they are basically, you're born with this. It can be quite severe. It can result in let's say, developmental problems, congenital abnormalities, etc. And because of that, it's actually very unlikely that this has an effect on fitness. So children with these diseases will unlikely reproduce, whereas healthy brothers or sisters will. So that means that quite a large proportion of genetic disease actually occurs sporadic. And that has for a long time challenged us geneticists, because you couldn't do family studies, you couldn't do association studies. So how could you find the genetic causes of disease? But another thing you could think about is actually to think about how can actually these kind of sporadic diseases, how can they occur, and if they are, let's say, impacting fitness, how can they still occur frequently in the population? Because if you think about, for example, intellectual disability, if you talk about an IQ below 50, that actually occurs in more than 0.5% of the population. So it is actually quite frequent, but it occurs mostly sporadic. You have one child in the family who has the severe intellectual disability. The parents have a normal IQ, and brothers and siblings have a normal IQ as well. So the question is very much, well, first of all, how can we identify these genetic causes underlying these sporadic diseases? But also the question that people had is, how can actually these reproductively lethal disorders, as you could call them, occur so frequently in the population? So for that, you have to do genetics, and that's what we've done. So I did a postdoc in San Francisco, as Claudia mentioned, and there I learned to work with genomic microarrays in the end of the last millennium. And basically I came back to the Netherlands to set up these microarrays, consisting mostly of back clones, and later we used SNP arrays from companies like Affymetrix, to look at deletions and duplications in the genome. And we've used that in many different ways to find the causes of rare diseases. And one of the advantages that I had when I moved to Nijmegen from America is that there was an excellent clinical group there, and they had collected DNA from many syndromes which the genetic cause had not been identified. So one of these syndromes was called CHARGE syndrome, which is an acronym, a combination of congenital heart abnormalities, renal abnormalities. So a very severe disorder, quite common for a syndrome at least, and the cause was unknown. And so what we did, we basically hybridized the DNA of a number of these patients to our microarrays, which were state of the art at that moment. They contained about 3,000 clones all over the genome. And we were lucky to find a deletion in one of these patients on chromosome 8. And we by also looking at the parental DNAs, we found that that deletion occurred de novo. So it was present in the patient, but not present in the parents. And that pointed us to the fact, like, well, okay, this may be the location for this syndrome. So this is where, like, in a way, it's a traditional positional cloning approach. So we identified now a region on chromosome 8 in a patient with CHARGE syndrome. And of course, we thought, well, probably the gene that causes CHARGE syndrome when mutated or deleted is located in this region. We had DNA from another 15 patients with this syndrome. They did not have deletions. What we in the end decided to do is basically sequence all the genes located in the region on chromosome 8q12. And to make a long story short, in the end we found mutations in one gene that we called chromodomain 7. It was a new gene, and that basically caused CHARGE syndrome, and it occurs de novo in all patients. So parents don't have these mutations, but the children do. And then we have also some patients with deletions in this deleting this gene. So that was one approach which allowed us to find a gene for a important syndrome. But that's, yeah, quite rare that you do find like that. So we had also access to a much larger cohort of children with non-syndromic intellectual disability, and that's basically the largest cohort of children that you see in the clinic. So we started to look at these with our microarrays, and this is with an Affymetrix array. And you see here a significant deletion, quite large. And what was very standard in the cytogenetics field is that if you would find a deletion like that, you would always look at the parental samples to make sure it's not a benign variation that's just inherited, because that could of course be the cause. But the fact that in this case, again, the parents do not cause this deletion, as you see here on the right, points to a very likely causal role for these kind of deletions in disease. So in this case, again, this is a de novo deletion that likely causes intellectual disability. So these deletions, however, in non-syndromic intellectual disability, these deletions and duplications can occur all over the genome. And that's what you see here. And we were involved in basically implementing microarrays in the diagnostic setting in about 2005. As one of the first labs in the world. And yeah, we could show that de novo deletions and duplications explain about 15% of intellectual disability. So you see they can occur all over the genome, but sometimes there's regions where there is multiple patients with the same deletion or overlapping deletion. An example you can see here on chromosome 17, and this was an interesting region on chromosome 17 Q21.31, where we found a number of de novo deletions, basically with the start and the end of these deletions more or less overlapping. We noticed from the mechanistic point of view that these deletions were close to a low copy repeat, and basically it turned out that you would have that the parents of these children, they always had a predisposing inversion, which actually basically positioned the low copy repeats in the right direction. So there could be non-allelic homologous recombination resulting in these kind of deletions. And then from a clinical point of view, our clinicians identified, apart from the intellectual disability, significant hypotonia and characteristics facial features. So that was an interesting observation. It was made by a number of groups around the world at the same time, so clearly quite frequent. And of course, then the question is, what is the causal gene in this region? And for that, we basically had to wait a number of years until we found a few patients with the same kind of clinical features who either had a partial deletion. So you should see two patients here with a partial deletion, so a smaller deletion, which of course could be used to fine map the genomic region. And then there were two patients who had mutations in a gene called CANCEL1, and that turned out to be the dosage-sensitive gene in this region of chromosome 17 that caused this syndrome. So again, a nice example of a deletion, a de novo deletion, that then points you to a genomic region. And then if you continue, you can find the causal gene. And importantly, again, for the story today is that all of these patients have de novo mutations or deletions. So again, that is very important cause for disease. Now, we know, of course, that de novo chromosomal abnormalities are extremely important. Down syndrome is a good example of that. We now have seen a number of these deletions. We've seen a number of point mutations. But what is also clear from a genomic point of view is that most de novo events will be point mutations. And for that, to really start looking into de novo mutations, you need to look at all point mutations occurring throughout the genome. So clearly for that, we needed something else than microarrays, and next generation sequencing came along, which of course is fantastic for looking at the genome and looking at mutations. So one of the first examples of where we used next generation sequencing to identify the genetic cause of an unexplained clinical syndrome is a syndrome called Schinzel-Giedion Syndrome. So again, in Nijmegen, we had access to DNA from a number of these patients. Again, this is a rare sporadic disorder, so no familiar cases. We had done microarrays in these cases and then had not identified any deletion or duplication there. So you're basically stuck until exome sequencing came along and later genome sequencing. And in this case, we decided to do exome sequencing for 4 patients with this syndrome. And our hypothesis was that these patients would all have a de novo mutation in the same gene, because it's a very characteristic syndrome, this Schinzel-Giedion syndrome. So we looked at the exome data. This was in 2010. This was basically one of our first exome sequencing experiments that we ever did. So we identified variants by exome sequencing. It's now very standard. So we had about 12,000 variants affecting the coding region. Of those, 5,500 affected the protein, and about 180 of those were absent from what we at that moment had as a control cohort. So basically the normal population. So that was great, because basically we did not know anything about the genetic cause of this syndrome, and now we had reduced this to about 180 genes. So that's not bad. But of course, the good news is that we had data from 4 patients, so we could combine them and basically look for overlapping genes. So basically, is there one gene in which all 4 patients have a rare protein affecting mutation. And indeed there was, because else I wouldn't have told this story. So in this case, we identified a mutation in the gene ZPP1, a rare predicted pathogenic mutation in this gene in all 4 patients. And importantly, again, for the story of today, is the parents did not carry this mutation. So the mutation occurred de novo in the germline of this child in all 4 patients. And this basically was the cause of the Schinzel-Giedion Syndrome, and it was the first dominant cause of a disease identified by exome sequencing. So that was great, and absolutely this has, of course, dramatically changed the way we do disease gene identification. Exome sequencing, we all now know, is fantastic for that. But it's, of course, also fantastic for looking at these kind of de novo mutations. Now, when you look at syndromes, it's relatively easy because you could say, well, I sequenced 2 or 3 or 4 patients with the same clinical syndrome and then try to find, try to look whether there is a single gene which is mutated in all 4 patients. But when you go to, again, the kind of non-syndromic, more common disorders like developmental delay, then we know for sure that these disorders will be genetically very heterogeneous. So sequencing 4 or 5 patients with intellectual disability in itself, that doesn't help you to prioritize variants because they will all have a causal mutation in a different gene. So what we started to do in 2010 for the first time was actually sequence not only the patient, but then also sequence the parents with that. And then the idea was that we would focus on de novo mutations, so there would be a drastically reduced number of variants to look at. And then, yeah, hopefully these de novo mutations would be good candidates for these disorders. So that's what we started to do, and we did a first pilot study in 2010. So this was the first time that patient-parent exome sequencing was done, which is now very routine. So very proud that we started this off basically. So we looked at 10 patients where we did exome sequencing, and you see here on the right, well, on the left, you see how we filtered down the variants. So again, we filtered for variants in the coding region that affected the protein that were not present in the normal population. And then we did some validation because, of course, there is still some sequencing artifacts sometimes. And in the beginning, perhaps if you don't sequence the parental samples deep enough, you may actually think that a variant occurs de novo, but it was simply missed from your father or your mother's DNA sequencing. So we had to do some validation. And in the end, we had between 0 and 2 de novo mutations in the coding region. So that affected different genes. You see that here on the right in these 10 patients. But interestingly for us is that out of the de novo mutations that we identified, 2 of them affected a known intellectual disability gene. Basically the symptoms that these patients had with these mutations overlap very much with what was known for patients with mutations in these genes. So basically, for these 2 patients, we could say this is a diagnosis. There was a 3rd patient who had a mutation in the gene called GERD1C. That was not de novo, it was actually inherited. Oops, but that was occurred on the X chromosome. And in this case, this was we actually then looked at the grandparents, and it turned out that the mutation occurred de novo in the mother, but then resulted in intellectual disability in her son. So this is, of course, often happening on the X chromosome. So that was really quite an important study because it really showed the power of trio-based exome sequencing. And it also showed to us that on the one hand, you can immediately make some diagnosis, but we also found quite some additional de novo mutations in very strong candidate genes for intellectual disability. But of course, that was just the first time that we saw a mutation in one of those genes. So we clearly needed to expand those studies. So basically, this pointed me for the rest of my life to my career. So I'm looking at de novo mutations. That's what I do. And basically, these mutations, of course, are critically important for evolution, right? Every variant that we ever inherited occurred de novo once. So it's critically important for evolution, but they're also, because they've not been selected through evolution, they can be an important cause of disease. So when we were doing this in disease, other colleagues, and notably in DECODE in Iceland, started to look at healthy populations, and they did genome sequencing for the first time in healthy parent-offspring trios, and they showed that every individual carries between 50 to 100 de novo mutations. So that's not a dramatic number if you think about the millions of variants that are inherited. But yeah, clearly from the work that I just showed already, it means that these mutations are actually quite important when it comes to disease. So when you think about that and you think of the role of de novo mutations in disease, it's important to know something about how often de novo mutations occur. So about 50 to 100 per individual. But it's also important to think about the mutational target of your disease. So with that, I mean, let's say, for example, with CHARGE syndrome, if you see here on the left, I just showed you that CHARGE syndrome is caused by de novo mutations in the gene CHD7. So it's really basically a single gene, mutations in that gene cause CHARGE syndrome. Now, when you have 50 to 100 de novo mutations in your genome and 1 to 2 in the coding region, the chance that one of those affects that gene is very small. So therefore, the disease will always occur quite infrequent, so it's very rare in the population, fortunately. However, when you think about a disorder like intellectual disability, where there could be thousands of genes, where one or, only when you have a mutation of one of these, it could be sufficient if it's disrupted to cause intellectual disability. And of course, you can imagine that actually intellectual disability can occur much more frequent and can actually be explained largely by de novo mutations. So that's the hypothesis we had. That definitely does mean that the disease is much more heterogeneous. So that has effect on the number of patients you need to sequence if you want to look at recurrent mutations, etc. But it does show that these de novo mutations can actually be quite important. So moving forward, we've done pilot studies where we started to do a genome sequencing in intellectual disability. And of course, that allows you to look also at the non-coding part of the genome and gives you a better insight in the structural variation. So one example I just want to show is a de novo duplication that we identified in a patient where we did genome sequencing. Now, of course, you could have identified these with microarrays, although this one was quite small, if I remember correctly, so it was missed with microarrays. But if you would see it with a microarray, you would say, okay, that's a duplication. You then go to the genome browser, you look at the genes, you think, well, these genes don't really explain intellectual disability. But importantly, of course, duplications, they can occur somewhere else in the genome. So one of the things we noticed in this patient, when we looked at the sequencing reads, that actually this duplication was not occurring in tandem, but actually moved to another chromosome. In this case, it moved from chromosome 4 to chromosome X. And when we did some further digging in our data and further validation, one of our computational scientists, Jane, did this work, Jane Heracqua. She noticed that the sequence on chromosome 4, actually, this is affecting a gene called TANM3, that actually inserted into a gene on chromosome X called IQ-SEC2. And when you study intellectual disability and you see that a gene starting with the term IQ is disrupted, then you get suspicious. And indeed, the IQ-SEC2 gene encodes for a gene that is basically involved in X-linked mental retardation, so in X-linked intellectual disability. So I think that was a great example of showing how genome sequencing is really important, not only to identify point mutations, but as the structural variation is, of course, also an important cause of disease. So we showed in this publication in 2014 that de novo mutations are actually the major cause of intellectual disability, severe intellectual disability, an IQ below 50. And we show that genome sequencing is the best test to do for these because it picks up, of course, not only these point mutations, but also copy number variations. So you see here on the pie chart that the large majority of causes is de novo single nucleotide variants, but also a considerable number of copy number variations. And the inherited recessive cause of intellectual disability is actually quite low in outbred populations. So there's still quite something to detect and to discover. So not all patients get a diagnosis at this stage with genome sequencing even done. Partly that is because there are still lots of mutations in new genes that have to be identified and characterized clinical. But of course, there's also other things such as the non-coding part of the genome. So one of the things that we and others have been involved in is trying to identify these novel intellectual disability genes. But because of the genetic heterogeneity that I already referred to, you need to really sequence a very large cohort of patients. And this is shown here by this figure here from Fitzgerald's, which basically shows that you need to sequence thousands of trios, patient-parent trios, before you find a significant enrichment, in this case of loss of function mutations. So these are the most severe mutations. And yeah, that's basically, you need tens of thousands of trios in order to identify all the genes. And of course, that's critically important, because once you've really identified them as being significantly, let's say recurrently mutated, you can then only implement these genes as targets in your diagnostics. So just some additional studies that have been published over the years, for example, in congenital heart disease, looking at 1,200 patients, identifying 21 recurrent mutated, de novo mutated genes. A study looking at autism, 6,000 patients, identifying 49 genes. And then a study from Kaplanis from 2020, looking at 31,000 patients with developmental delay and their parents, and identifying more than 280 developmental disorder genes and many of these new ones. So clearly showing that we need to work together, share data with each other in order to find all these genes and improve the diagnosis for these patient groups. So that's not only developmental delay, that's autism, that's congenital heart disease, etc., etc. With colleagues in Nijmegen, we've been looking at smart, let's say, ways to look at the data and basically mine these data in a different way than just saying how many recurrent mutations do we have. So one example here is from one of my colleagues, showing that you sometimes see a clustering of mutations in a functional domain, and that in itself can already allow you to identify, let's say, significant enrichment of mutations compared to controls in a lower number of patients. For the gene PAX2 there below, you see that all de novo mutations occur at the same position in the gene, pointing to a gain of function mutation in this case. So there's lots of things that you can do to further improve your data analysis. But of course, and this is from a recent work, and there have been lots of papers just in the last month showing the importance of improved sequencing, but also improved alignment of our sequencing reads to a better reference genome. Is just showing what you can do if you want to detect all de novo mutations in a genome. So in this case, these colleagues first did standard Illumina sequencing, short read sequencing, and 2 siblings, identified 142 de novo mutations, and added long read sequencing, so basically expanded the number of de novo mutations to 171, simply because you can then identify mutations in regions that were missed with short read sequencing. But then moved to a more complete reference genome, the telomere to telomere reference. And actually that further improved their number of true de novo mutations to 195. So that shows you that there is clearly a lot of, let's say, improvements to be made. Yeah, I'm here referring to Nurk et al. From bioRxiv, but I think that's just out in Nature. Importantly, they also show that you can identify de novo repeat expansions and de novo structure variations. So perhaps at a lower frequency, but still these can be detected. So I think when you want to study de novo mutations and their role in disease, there is still a lot of work to be done in basically in better sequencing with long read sequencing, and then indeed in better alignment to a more complete reference genome. All right. Now, of course, de novo mutation in the coding region is one part, but then it's there is the rest of the genome, which is about 98%. And of course, that is, again, where a lot of us are very interested in, but we also realize the challenges because it's an extremely large part of the genome. And of course, again, the question is, can you find recurrently mutated non-coding mutations that are really significantly associated with disease? So far, we have not ventured into that. That will require, again, enormously large numbers of patients. But we were in one way, we were lucky with one example that I just wanted to show you here. So in this case, we did genome sequencing in a patient with epilepsy and psychomotor delay. For this patient, the clinician thought of a GLUT1 deficiency, had already asked for sequencing of the known genes in this disorder. SLC2A1 is one of the major ones, so I'll just highlight that and we'll come back to that. Microarrays were done, exome sequencing was done. Oops, sorry, I'm going too quickly here. A few de novo mutations were identified, but they did not match to the phenotype. So basically, genome sequencing was done. And well, in this case, because there was such a clear deficiency already identified, we looked at, we identified the de novo mutation in the 5' UTR of SLC2A1, the gene causing GLUT1 deficiency. And in this case, that resulted in a novel start site leading to a premature stop codon. So in this case, yes, you can say it's a non-coding mutation, but that is indeed because we were so close to already having a diagnosis. So that is not completely fair. And that, of course, it will be much more challenging if you don't know what to look for. All right. So that brings me to a little bit of a conclusion before I move on to a second part of my talk. So clearly, inherited variation is very frequent. We have millions of variations. They have been selected in evolution, and they are not likely to cause early onset dominant disease. Whereas on the other hand, de novo mutations, we have very few, but they have not undergone extensive selection, and they actually explain a major fraction of early onset dominant disease. And of course, if they're benign or advantageous, they will become inherited variation in the next generation. So if these de novo mutations are so important in disease, it's important to learn a little bit more about where did they come from, how often do they occur, and you know, can we perhaps reduce the number of de novo mutations in our offspring? So, well, basically they can occur at different stages. They can occur during development of the germline in the paternal and maternal genome, but also with paternal and maternal aging, and they can occur early in the embryonic development of the offspring. Well, one of the things that we know is that, importantly, a risk factor is parental age at childbirth. And here you see a graph where you see that women are very smart, as we know, much smarter than us. They stop reproducing at a certain period, but males, they think that they can continue into their 70s. Now, this is a logarithmic scale, so it's not that bad. But we have to be a bit careful. And here you see a few famous men. And this is not the number above there is not the age when they died, but the age when they got their last child. So clearly that was always something we were proud of older men getting children, whereas with women, we always say, be careful, you have to stop reproducing. However, when we started looking at de novo mutations, and I already referred to this publication from DCODE, from Augustine Kong, they showed for the first time that the number of de novo mutations is directly associated to the age of the father at conception. So basically, younger fathers will get children with much less de novo mutations than older fathers. And you see here, it's almost linear between 20 and 40. And you see that about 80% of mutations have a paternal origin. So it's clearly most de novo mutations come from the father. Together with a few other groups, we've looked into this ourselves, and we showed indeed we could, of course, replicate this paternal age effect. So basically showing that with every year a father gets older, he passes on one additional de novo mutation to his child, whereas with every age a mother gets older, there is also a maternal age effect, but that's much less strong. There will be about a quarter de novo mutation per year added. So that's for you all to know. And we just recently published this pilot study where we looked at, we basically asked the question that there's now lots of children that are born through assisted reproductive technologies. And we were wondering whether children born with IVF or ICSI whether they have more de novo mutations in their genome. So we've done a relatively small pilot study. So you see it here. We had 18 patients with excities, 17 with IVF, and 18 spontaneous pregnancies. We basically, because we knew already of the paternal age effect, we decided to focus on groups basically with young parents, young fathers, and a group with old fathers in each of these. So that further makes the groups even smaller. But then we did genome sequencing. We did for a large majority of these children, we did genome sequencing twice in both the children as well as their parents. And on the right, you basically see the outcome. So you see from left to right, you see the spontaneous children with spontaneous pregnancy. And you see the number of de novo mutations in the group where they have either a young father or an older father. And you see clearly there is a paternal age effect in the spontaneous group, but also in the IVF and in the excities group. But then when you start comparing amongst these three groups, we did not see a significant increase in the number of de novo mutations, and also not a significant decrease, I should add, because it looks like there is a little bit of a decrease, but that's not significant. So basically, we see no significant difference in the number of de novo mutations for children born with the different methods of conception. Now that needs to be further replicated in much larger cohorts. There could be absolutely more subtle effects, but at least we can exclude that there is a major impact of assisted reproductive technologies on the de novo mutation rate, which of course is good news because more and more children are born this way. Now that brings me to last topic of my talk, and that is talking about de novo mutations and male infertility. So when I moved to Newcastle a few years ago, I already started to focus on another disease because I thought everybody is now doing this developmental delay work and I can do something else. And I was interested in studying infertility, male infertility in particular, because there was very little known about it, very little known about the genetics of this. And of course, you could imagine that when you're infertile, well, you probably did not get that from your parents. Your parents were both fertile. So again, here, de novo mutations may be an interesting explanation. So infertility is basically defined as a failure to achieve a pregnancy after 12 months of regular unprotected sexual intercourse. And it is basically, it can happen both from a female and a male side. And for research, it's easier to focus on the male, I have to be honest, because we can basically look at the ejaculate and diagnose what's happening. So we, in our research, focus both on quantitative and qualitative sperm defects. But in this study, I'll talk mostly about quantitative sperm defects. So we basically look at men that produce no sperm in their ejaculate, or they produce a very low number of sperm in their ejaculate. So they're severe oligospermic or azospermic. There's many different explanations, but actually, to be honest, for the majority of cases, we don't know what's happening. And one reason why we don't know is because genetics is still, let's say, not really done in this field. It's basically at least a decade behind what's happening now in most other diseases, of course, depending on the country that you live and work in. Of course, karyotyping is being done in infertility. There has been the discovery of the CFTR gene, which is an important gene for infertility. So that has been that's looked at. And then there are microdeletions on the Y chromosome that were identified in 1995 that have been implemented in diagnostics. But basically, since then, nothing happened. And still at present, even the guidelines talk about doing karyotyping and looking perhaps for the androgen gene as another gene to look at, but that's about it. So clearly, not a lot is happening if you compare that to children with developmental delay, who here in the UK, they basically get genome sequencing as a standard point of care at this moment. Now, for male infertility, as I said, I was interested in the role for de novo mutations. And based on what we know, we can actually say there is already quite some evidence for that. So Klinefelter syndrome, the XXY, is causing infertility, and that's of course a de novo chromosomal abnormality, but also microdeletions on the Y chromosome in the AZF region occur de novo. So again, these abnormalities, which are amongst the most frequent causes of male infertility known, they both occur de novo. But what about de novo point mutations? Because of course we know that these will probably be more important than the chromosomal abnormalities. Well, actually, there's basically one publication from David Page and his group in 1999 showing a de novo mutation in a gene on the Y chromosome called USP9Y. That publication has been challenged. It's not clear whether de novo mutations in that gene are immediately causal for infertility, but basically nothing has happened. Nobody has looked at de novo mutations in infertility. So that was until recently. So we just published this paper and Claudia, of course, is very prominently among the authors, and I thank her again for collaboration. And I'll explain a little bit what we did in this study. So I just talked to you about intellectual disability and other diseases where people are now looking at tens of thousands of trios. This is a bit more challenging in the field of infertility. In developmental delay, of course, one of the advantages is that this is now routine diagnostics, but it's also parents are coming with their child to the clinic because their child is not developing. In infertility, for our research, it's very sad, but infertile men don't come to the clinic with their parents. And that, of course, makes it much more hard for us to, much more difficult for us to do these kind of trio analysis. So we basically have to ask these patients whether they want to collaborate with this research and whether they are okay to involve their parents into the research. And infertility is in many countries still a taboo, so it's not that easy. Parents often don't know that their son is infertile. So we have to overcome quite some struggles to get basically the material that we need for our research. So in this study, we now managed to get 186 patient-parent trios where we have blood samples from the patients and saliva samples from the parents. And we performed exome sequencing basically as we always did. So we identified lots of de novo mutations, as you would expect, very much in line with the numbers that you would see in the normal population. So there's no increase in the number of de novo mutations. But of course, that's also not what we expected. We really want to see are there specific genes affected. So one of the first de novo mutations we identified occurred in a gene called ODF1, outer dense fiber of sperm tails. So I thought when I saw that, now that's great, that must be the cause of infertility. So this research is not going to be too difficult. Perhaps it's not that genetically heterogeneous. But unfortunately, that's the only patient with a mutation in this gene that we found. And basically, all of our mutations occurred in different genes. So we found no de novo mutations in known infertility genes. But then again, there is only 4 dominant infertility genes known. So there's very few known so far. So basically, we need to do this research in order to find these genes. And then there were no genes with recurrent mutations. But again, as I mentioned already, we have only 186 trios. And if you think about the genetic heterogeneity, we know that there is 10,000s of genes expressed during the spermatogenesis. So it can be extremely genetically heterogeneous, similar to developmental delay. Then indeed, this cohort is basically too small. So, so what we did then is we classified our mutations based on predicted severity. That's all the kind of things that you would normally do. So we said, okay, it looks like we may have a number of mutations that could be causative of the infertility. But what can we see more? So one of the things we noticed is that we saw quite a lot of loss of function mutations, so frame shift and stop gain mutations. And when we looked at the genes that they affected, we noticed that the genes had a very high PLI score, which is a score basically indicating the tolerance to loss of function mutations, as you may know. And so we could show that there is indeed an enrichment of de novo loss of function mutation in loss of function intolerant genes in our cohort. So that's an interesting observation. But of course, most de novo mutations will be missense mutations in the coding region. And there is also a score for, let's say, a tolerance of a gene to missense mutations, and that's called the missense Z score. And we could show that there was actually an enrichment of pathogenic de novo mutations in missense intolerant genes. So that was, again, an interesting statistical observation in our data. Now, when we looked at the mutations predicted to be pathogenic, and we then did some standard analysis to just look at some pathways, we noticed one pathway affected, and that is mRNA splicing. So via the spliceosome, so you see here a number of genes where, we noticed the de novo mutations predicted to be pathogenic. So that was an interesting observation because the spliceosome is quite important in spermatogenesis. So that's something we wanted to further look into. But our challenge was that we were the first to do this in a trio cohort, and that makes it difficult to do any sort of replication. So it's nice to be the first to do something, but of course you can't check then and say, let's get some other cohorts from others. So therefore we collaborated with various groups, including colleagues from the International Male Infertility Genomics Consortium that we founded. And we basically got in total, we got exome data from 2,500 infertile men. But in this case, we don't have the trio data. So we just have exome data of the infertile men. And the question we asked is, we had these genes with de novo mutations. Would there be predicted pathogenic mutations in these genes in infertile men, more so than in fertile men? And we had a cohort of fertile Dutch men and Dutch women. Basically, these were the parents of children with intellectual disability. So we could basically compare that. So we did a burden analysis, and as you can see this is the top list. So these are the most, let's say, significantly relevant genes. And in fact, if you look at the adjusted p-value, you see that it was just a single gene for which we identified significance. So this is the gene RBM5, which was actually on the previous slide as one of the genes involved in the spliceosome. And we identified 7 infertile men with a predicted pathogenic missense mutation in this gene and 0 fertile Dutch men. Now, there's another few genes. You see the gene UE1 with 6 mutations and 0 in the controls, but that was still not significant after the p-value. So it shows you that we need to have larger cohorts. But it looks like there is a number of interesting genes there, but RBM5 is the only one that reached significance. So finally, for our study, we were interested in one question. Of course, I already showed you that de novo mutations mostly occur on the paternal allele, about 80%. And in this case, it would be quite interesting to think about these de novo mutations causing perhaps male infertility, because if indeed they would occur on the paternal allele, it would mean that there would be a sperm produced with a de novo mutation in a fertile man, who is the father then of what becomes an infertile patient. So basically the question is, can you get sperm viable which carries a de novo mutation that actually causes infertility in the next generation? So for that, we did long read sequencing to phase de novo mutations and to check whether indeed whether there was a paternal or maternal origin of the de novo mutations. So in total, we did that for about 29% of the mutations. We didn't do it for everything. But what you can see here is from this graph, we basically have these mutations classified as either possible causative, unclear, unlikely causative, or not causative. And you see that there is a significant number of possibly causative mutations which actually are paternal, as well as the unclear ones, which are also 5 out of 11 are paternal. So it looks like indeed, if indeed it turns out that these mutations are truly causing male infertility, then we also show that they mostly occur still on the paternal allele, as we would expect that for normal de novo mutations to happen. So the question is, how can that happen? So we have a few possible explanations for that. So one thing is that it could be that the de novo mutation occurs in a gene after the developmental window in which this gene is active. So basically, the gene plays a role earlier on in spermatogenesis. That could be. And then, of course, it doesn't really, there's not a problem for this de novo mutation to occur. At least in the father. then it could be that the novel mutation affects a gene that actually is critical for somatic cells supporting the future germline. So it's not really affecting the germline itself, but it's more important that that gene plays a role in the somatic cells. But another explanation, and that's quite interesting, is that from spermatogonia onwards, male germ cells, they form cysts and they share mRNA and proteins. So perhaps actually these mutations occur, but the sperm cells don't mind this because they actually get RNA and protein from other spermatogonia, and therefore can still fertilize. So that would be indeed masking the detrimental effect of the novel mutations. All right, so a lot of interesting biology there as well. So finally, what are we going to continue to do? We would like to extend, of course, this trio-based exome sequencing, and we are collaborating with colleagues from around the globe. So if colleagues in Mexico are interested, please help us out, because we need many more patient-parent trios. So happy to discuss. We have just finished genome sequencing all these about 200 trios. So we moved from exome to genome sequencing. So we're looking now at structural genomic variation, repeats, expansions, and non-coding mutations, as you can imagine. We're doing some functional studies in genes like this RBM5 gene and other splicing genes. And we try to work with colleagues in the clinic to implement genomics in male fertility diagnostics. But another thing we are doing, which is more a small scale, is continuing some work with the assisted reproductive technologies, because we also have access to DNA of basically boys who were born to infertile men through the use of assisted reproductive technologies. Some of these boys have lower sperm counts, and we're basically sequencing their genome to see if they indeed have a genetic cause which is inherited from their father. And that's, again, I think important to show the world that you can inherit infertility through assisted reproductive technologies, which is, of course, as you would imagine. What's next for this field of de novo mutation studies? I think there is a lot of things happening. I think clearly there is much to do still in the detection and phasing of de novo mutations with let's say, these new reference genomes with long-read sequencing. Repeat expansions and structure variations are critically important to look at. We need larger cohorts for the detection of these mutations in general, especially when you want to go to the non-coding part of the genome, which is a challenge. Then one of the things that people are looking at is looking at the recurrence risk of de novo mutations in the clinic. It's not the case that if you have a de novo mutation that your recurrence risk is completely zero, because sometimes these de novo mutations actually occur earlier on. So lots of work to be done in this field. As I always like to say, we only just started. So that's the best thing for a scientist, of course. So let me thank finally lots of colleagues from around the world, and including Claudia and Chris. I want to thank lots of colleagues from Radboud University, from Newcastle, colleagues from the International Male Fertility Genomics Consortium, and lots of funders and lots of colleagues, as you see here from pictures, with whom I've had a lot of fun. So thank you very much for your attention. And I'm gonna, oops, let me go back, because that's like what we need. Let me just finish here, and thank you very much.
Thank you very much, Joris.